Hello friends, welcome to our YouTube channel SRIP EduFarm. In this video, I have described important mechanism of transmembrane signaling in simple way that will give an idea about the mechanism by which number of drugs act through. So let's start the topic. Now mechanism for transmembrane signaling. Most membrane signaling is accomplished by a small number of different molecular mechanisms, and each type of mechanism has been adopted through the evolution of distinctive protein families to transduce many different signals. These protein families includes receptor on cell surface and within the cells, as well as enzymes and other components that generate, amplify, coordinate and terminate the post-receptor signaling by second messenger in the cytoplasm. There are five basic mechanisms of transmembrane signaling. First is intracellular receptor for lipid-soluble agents, in which lipid-soluble ligands crosses the membrane and act on intracellular receptors. Second is ligand-regulated transmembrane enzymes, in which a transmembrane receptor protein whose intracellular enzymatic activity is allosterically regulated by a ligand that binds to a site on the protein extracellular domain. Third mechanism is through cytokine receptors, where a transmembrane receptor that binds and stimulates a protein tyrosine kinase. Fourth mechanism of signaling is through ligand-gated channels, in which a ligand-gated transmembrane ion channels can be induced to open or closed by binding of a ligand. Last basic mechanism is through G protein and second messengers, where a transmembrane receptor proteins stimulate a GTP binding signal transducer protein, also called as G protein, which in turn modulates production of an intracellular second messenger. Now let me describe each one by one with example. First is intracellular receptor for lipid soluble agent. Several biologic ligands are sufficiently lipid soluble to cross plasma membrane and act on intracellular receptors. One class of such ligands includes steroids like corticosteroid, mineralocorticoids, sex steroids, vitamin D, and also thyroid hormone, whose receptor stimulate the transcription of the genes by binding to the specific DNA sequence near the genes whose expression is to be regulated. As an example, mechanism of glucocorticoid action is explained. The glucocorticoid receptors has three distinct domains. A heat shock protein HSP90 binds to the receptor in absence of hormones and prevent folding into the active conformation of the receptor. Binding of a hormone ligand causes dissociation of HSP90 and permits conversion to the active configuration. This mechanism of signaling has two therapeutically important consequences. First, all of the hormones produces their effect after a characteristic lag period of 30 minutes to several hours for the synthesis of new proteins. And second is, the effect of these agents can persist for hours or days after the agonist concentration has been reduced to zero. Second is ligand-regulated transmembrane enzymes, including receptor tyrosine kinase. This class of receptor molecule mediates the first step in signaling by insulin, epidermal growth factor, transforming growth factor, and many other trophic hormones. As an example, mechanism of activation of the epidermal growth factor receptor is explained. The receptor polypeptide has extracellular and cytoplasmic domains. Upon binding of EGF, the receptor converts from its inactive monomeric state to an active dimeric state, in which two receptor polypeptides bind non-covalently. The cytoplasmic domains becomes phosphorylated on specific tyrosine residue and their enzymatic activities are activated. Third mechanism of signaling is through cytokine receptors. Cytokine receptors respond to a heterogeneous group of peptide ligands that include growth hormones, erythropoietin, interferons, and other regulators of growth and differentiation. After activation by an appropriate ligand, separate mobile protein tyrosine kinase molecule are activated. resulting in phosphorylation of signal transducers and activators of transcription, that is STAT molecules. STAT dimers then travel to the nucleus where they regulate transcription. Next is ligand-gated ion channels. Many of the most useful drugs in clinical medicine act by mimicking or blocking the action of endogenous ligands that regulate the flow of ions through plasma membrane channels. The natural ligand includes acetylcholine, serotonin, GABA, and glutamates. As example, the nicotinic acetylcholine receptor, that is a ligand-gated ion channel, is explained. It is composed of 5 subunits, that is 2 alpha, 1 beta, 1 gamma, and 1 delta. The receptor opens a central transmembrane ion channel when ACH binds to the site on alpha subunit. Last mechanism of signaling is through G protein and second messengers. Many extracellular ligands act by increasing the intracellular concentration of second messengers such as CMP, calcium ion, or the phosphoinositides. In most cases, they uses a transmembrane signaling system with three separate components. First, the extracellular ligand is specifically detected by a cell surface receptors. Then, the receptor in turn triggers the activation of G protein located on the cytoplasmic face of the plasma membrane. Then, the activated G protein changes the activity of an effector element, usually an enzyme or ion channel. This element then changes the concentration of the intracellular second messenger. For CAMP, the effector enzyme is adenylyl cyclase. Now, all the basic mechanism of transmembrane signaling has been explained now in short. Thank you so much.